Sorry, I
Good
didn't get that.
afternoon. See, I'm just changing slide. Just to confirm, we are not going to face the technical glitch what we have faced yesterday. So please let me know whether you are going to get this slides or not. I'm just mentioning something on the slide and you please let me know whether you are able to see it or not. I'm just marking slides. I'm just marking slides. Slide number 1, slide number 2. Please let me know, can you get these numbers or not? Just let me know whether you can see my so you all are most welcome in this series of science and technology. So let me continue. We have so many questions. There's so many questions are scrolling on the screen. But before taking these questions, let me complete the topic what we started and what we were in the mid of the topic. So let me confirm and then I am going to take your questions separately. Fine. So see, the last discussion was all about, we were talking about all about the immunity. Right? The last discussion was all about the immunity. And in this immunity, we have mentioned the two different categories of the immunity. Like the first is innate and the second is acquired. Fine. We have discussed about this innate immunity. We have discussed about this innate immunity as a physical immunity, as a chemical immunity, as a cellular immunity, and as a viral immunity. Right? So there was a question, I'm not able to recall, there was a question from Ayush. See, when we talk about this physical, chemical and the cellular immunity, this physical immunity is providing immunity against all pathogens. Right? Means this physical, this chemical and the cellular immunity is against living pathogen. But when we talk about the viruses are living chemical, right? Viruses are living chemical, and against viruses we do have a provision of a special antibody, and this special antibody already we have discussed. I'm just repeating the previous discussion just to make because this question was asked by so many people. So I'm just repeating the previous discussion and see here just for a couple of the minutes so that you feel comfortable, right? See, this is the special antibody, and this special antibody is effective only against viral protein, and this is what? It's an interferon. Right? So when we talk about the interferon is a special it's a special kind of antibody, and this special kind of antibody is effective only against video is going. Are you getting my voice properly? Are you getting my audio and visual signals properly? Please confirm everyone. Please confirm everyone. Please let me know. Are you getting my video or not? Talk about coronavirus, whether we talk about hepatitis, whether we talk about any viral infection, right? We are prone to any kind of infection, and against infection, body starts responding in the term of antibodies. In the term of antibody, you heard about nowadays we are talking about the amount of antibody. Nowadays we are talking about the amount of antibody means after 28 days this plasma has enough amount of antibodies, right? Plasma has enough. Sorry. So nowadays we are talking about, see, after the infection of the corona, we are going to get immunity. So what is the meaning of this immunity? After the infection of corona, if someone is going to attain immunity, it means now that infected person is going to attain immunity after the development of the antibody, and this antibody is nothing but this is the interferon. So this time we are talking about the plasma therapy, means anyone who has recovered against corona, right? That individual has that the plasma of that individual has the sufficient amount of this antibody, and this antibody is quite effective only against this coronavirus. And technically, the nature of this antibody is what? It's an interferon. So when we talk about the interferon is a special kind of antibody which is effective only against viral infection. Is the logic behind this. Interferon is called as first line of defense against viral infection, right? That already we have discussed, right? So this viral immunity is all about the interferon. This viral immunity is all about the interferon. This viral immunity is all about interferon, which is a type of antibody, and this is first line of defense against viral infection, which is the first line of defense against viral infection, right? This is all about the viral immunity. While this cellular immunity, right? So when we talk about this cellular immunity, clear? This cellular immunity is because of the lymphocytes, and this lymphocyte is active against all living pathogens. It is active against all living pathogens. So all living pathogen means what? This could be the bacteria, this could be the fungus, this could be worms, this could be even the protistans like paramecium, like Entamoeba histolytica. So you heard about WBC. Lymphocyte is what? Lymphocyte is the type of WBC. And the WBC is what? WBC is the soldier system of body. the soldier system of the body that make a sense of it fights against all infection. And basically the nature of this infection would be living infection. Primarily it is responsible for protecting yourself against the all living pathogenic infection that make a great sense of cellular immunity. So cellular immunity is what? It's a wide perspective. Cellular immunity is applicable in the wide term, means it is required for all pathogens. While when we talk about the viral immunity, this viral immunity is applicable only for the viral infection. Means the cellular immunity is broad perspective, while the viral immunity is narrow perspective, which is restricted only for the which provides you immunity only against viral infection, while the cellular immunity is protecting you against all living pathogenic infection. Fine? Okay? Right. Fine? After doing this entire exercise, we started discussing this acquired immunity. Clear? And this acquired immunity that already we have done. I am just repeating. And this acquired immunity is what? This acquired immunity is immunity after birth. Clear? So immunity after birth, and in this category we have discussed about the two different categories. This acquired immunity, we have discussed two different categories. One is passive, that already we have done. Clear? And this is nothing but it's a colostrum, which is transfer from mother to newly born. Right? Mother to newly born, and we have discussed, because the mother milk is also good source of, apart from the nutrition, mother milk is also good source of the antibody, and this antibody is the IgA, that is the immunoglobulin A, that already we have mentioned in the previous discussion. It's the IgA, that is the immunoglobulin A. Clear? And the second is active immunity. This active immunity is permanent, while this passive immunity is temporary. This passive immunity is temporary, while active immunity is permanent. And in this category of passive active immunity, we have discussed two different approaches. Like one is natural and the second is induced. This natural is all about herd immunity, right? Nowadays you heard about asymptomatic corona, right? Probably there are so many people, the major chunk of the people, they are suffering from the corona, but hardly they are not able to see the symptom. So internally they are carrying coronavirus, fine, but see, they are good enough in their immunity. And after the infection of coronavirus, their body start responding in the term of corresponding antibody. Clear? So they are not able to feel the symptom of the corona, rather they are suffering from the corona, but they are not able to see the symptom of the corona because they are good in immunity. And this is what, it's a best example of the herd immunity. Right? Fine. So it is not applicable only in the case of corona. This we could apply anywhere in so many cases. Like see example, when we talk about the transition of weather from summer to winter, from winter to summer, we could see a number of cases. Number of cases, especially majority of the population suffering from the common cold, flu, fever and all. But even you could also see there are so many people, they are least impacted by this weather change, because weather changing is what? Weather changing that ensures the good temperature condition, and the good temperature condition is what? Whether we talk about summer to winter or even winter to summer, that particular period consists of where in that particular period the atmospheric temperature is somewhere, it's a 20 to 25 degrees Celsius temperature, right? So that temperature is best suited temperature for multiplication of any pathogen. And the reason is what? In the case of the during the weather changing, because of the suitable atmospheric temperature, the pathogen starts multiplying, and as a result of the abundance of the pathogen, the population usually starts suffering from the infection, and that infection is common as a common cold, as a flu, and all and all. But even at the same time, you could also see there are so many people, they are least impacted or they are not impacted by this pathogenic infection. Even they are prone to they are prone to the infection. Why? Because they are well enough, right? They have good amount of immunity, means antibody inside, means the body start responding well against the pathogenic infection. And as a result of the response of the body, right, they are able to defeat pathogenic invasion, and this is the reason behind they are good enough in, right, means they are good in internal immunity, and that internal immunity is nothing but it's a good amount, it's a classical example of the hard immunity. So I acquired hard immunity against any kind of the contamination without any medication or without any vaccination. That is what? That is the best example of the naturally acquired immunity, and that immunity is going to be sustained. That immunity is going to be sustained. That's why nowadays we are talking about naturopathy to improve. We are talking about the naturopathy, and intention of the naturopathy is what? Through the natural process, we are supposed to improve immunity. It means we are just talking about strengthening of internal immunity, and the strengthening of internal immunity is nothing but it is all about the herd immunity. Right? Fine? So this is what nothing but it's a herd immunity. Clear? And the second is what? It's a induced immunity, and this induced immunity is what? This is induced by vaccination process, and this is what? It's a vaccine. Ashish, there's a question from the Ashish. See, Siddhant, let me take Siddhant. Is herd immunity is same as the viral immunity? See, viral immunity is specific against the viral infection, but when we talk about the herd immunity, this herd immunity is broad in sense. Means this herd immunity provides immunity against viral infection. This provides immunity against bacterial infection. This provides immunity against any kind of the infection. Just like example, let me make you understand one example. Especially suppose in extreme summer conditions, right? If you are going to take a food from the hotel, so you and your fellow, or you and your friend, if you both are going to take food from the hotel, and probably suppose, and if this food is contaminated, so there could be so many chances. Like suppose if you both are going to be alright, you both are least impacted or not impacted by after taking even the contaminated food. Clear? Or even the second case is what? Your friend is suffering from the contamination like the loose motion, but you are well enough. And the third condition is what? Probably you both start suffering from the contamination. So the second condition is what? Out of both, means you both are going to take the contaminated food, right? But only out of these two, only one person is going to be affected against the infection. It means what? That individual has, the immunity of that individual is weak. Means that individual has weak herd immunity, while the second one is not impacted or least impacted even after taking the contaminated food, has a good herd immunity. So the herd immunity is immunity against any kind of the contamination, any kind of the pathogen, while the viral immunity is specific only against the viral infection. That is the basic difference between the herd immunity and the viral immunity. Right? Fine. So we can say like viral immunity is a subset of herd immunity. Herd immunity term is applicable for immunity against all kind of the pathogen, while viral immunity is effective only against the viral infection. Right? I hope you understand. Siddhant, Fine. So coming to this vaccine, this is permanent solution for the active immunity. That this time we are talking about the vaccine, especially in the case of coronavirus. We have two different types of indigenous vaccine, as a Covaxin and a Covishield, that I am coming to this point. So first of all, when we talk about the vaccine, right? When I talk about the vaccine, what actually the vaccine is? See here. See, when I talk about this vaccine, let me justify what vaccine is. This is what actually the vaccine is. Vaccine is the biological preparation that provide active acquired immunity to a particular infectious disease. Right? Particular infectious disease. Like example, you heard about DPT vaccine. This provides immunity against DPT. D means diphtheria, P means pertussis, T means tetanus, right? Then OPV, IPV, that is the oral polio vaccine or injectable polio vaccine. That is what? It's against polio, right? Then Covaxin or Covishield, this is against SARS-CoV-2, right? So I'm just taking example again. Hib vaccine is specific against Haemophilus influenza type B. So the point is that here vaccine is the biological preparation that provides active acquired immunity to a particular infectious disease, and vaccine is typically antigenic in nature. It's antigenic in nature. Vaccine is antigenic in nature. Any vaccine is antigenic in nature. Any vaccine is antigenic in nature, right? And the intention of vaccine is here. The intention of vaccine. See here. I'm going to elaborate this vaccine later on, but let me make you understand. This vaccine is basically antigenic in nature, and the intention of this vaccine is what? To stimulate antibody, and this antibody is required for immunity, right? So vaccine is basically stimulating agent, while antibody is what? It's a respondent, right? Vaccine is antigenic in nature and antibody is respondent in nature, and technically, chemically, both antigen and antibody, both are technically protein in nature. Both are technically protein in nature, right? So vaccine is nothing but vaccine is typically what? It's a pathogen. Vaccine is technically pathogen, right? It's a whole, either vaccine means what? Vaccine has, see, you see the difference between the contamination and the vaccination is what? It's a very simple difference. Contamination is always controlled. Contamination is always uncontrolled. But see what happened, we have captured that particular pathogen, then we have inactivated or killed this pathogen, Clear? And then the limited amount or the limited amount of complete pathogen or the part of this pathogen we are inserting in the body, that is nothing but vaccine. So the simple difference between contamination, and the vaccination is what? Contamination is always uncontrolled, and it is through the living pathogen. Contamination is because of the living pathogen, and vaccination is always controlled, and the vaccination involves inactive or dead pathogen, either completely or the part of pathogen, just to induce immunity in the term of responding antibodies. This is the basic difference between contamination, vaccination, and how vaccine works. So already it is mentioned here, a vaccine typically, you could see here, vaccine typically contains an agent, and this agent is what? It's a pathogen that resembles a disease causing microorganism, means pathogen. Clear? And is often made from the weakened and the killed form of microbes, or it's a toxin, or one of its surface protein. Yeah? You heard about the two different varieties of corona vaccine. One is Covaxin and the second is the Covishield. Right? Covaxin has killed form of coronavirus, while the Covaxin has only its surface protein. This is the basic difference between the Covaxin and Covishield. Right? So let me elaborate this particular point here. Clear? This is what? It's a coronavirus. This is coronavirus like this. And see what happened. We have simply inactivated this. Inactivated this, and then it is inserted whole pathogen. This is what? It's a Covaxin. Yeah? This is Covaxin. Clear? This is Covaxin, which is the joint venture of Serum Institute Pune plus AstraZeneca. Right? While in the case of Covishield, one of this spike protein, which is antigenic in nature. Here, whole pathogen is antigenic in nature. Yes, and then it is used for vaccine. Clear? And this is the concept of the Covishield. And this Covishield is the result of, it's a joint venture of Bharat Biotech, ICMR and NIV, Indian Council of Medical Research, right, and National Institute of Virology. Fine, okay. So coming back to this discussion, this part of the discussion, see here, look here. Let me check it out. Let me check it out. Probably we have just reversed. Just hold it. Hold it. Hold it. Sorry, we have just reversed it. Yes, sorry. We have just reversed it. Sorry. We have just reversed it. This Covaxin is the result of this Covaxin. We have just reversed it. Sorry, just corrected. This Covaxin is the result of the Bharat Biotech, and the Covishield is the product of, right, this is what, just make it, Covishield is the result of the Serum Institute of India and AstraZeneca Oxford. Clear? We have just reversed it, just corrected. Covaxin is the product of Bharat Biotech, ICMR and the NIV, while the Covishield is the product of the Serum Institute and AstraZeneca. I'm sorry, yeah, we have just reversed it, right. Clear? Fine. So coming back to this discussion, see here, see the purpose of the vaccine is what? The purpose of vaccine stimulates body immune system to recognize the agent, and here the body immune system is what? It's nothing but the antibody. It's the antibody to recognize agent as a threat or destroy it, and further recognize and destroy any kind of the microorganism, clear, associated with the agent. Means the body immune system is what? It's the antibodies, right? And the administration of the vaccine is called as the vaccination. And see here, the across the worldwide, now the WHO, now as per the WHO has reported licensed vaccine, means for the for the 25 different preventable diseases, license means WHO has provided licensed vaccine for the 25 different preventable diseases across the world. Clear? And you must be knowing about the smallpox. This is completely eradicated from the world. The polio is almost eradicated from the world except a few countries, and even the measles is also eradicated from the world except a few countries. Like the tetanus is also like the same, right? So we are not supposed to discuss about, see, the point is that here we are not supposed to discuss about so much about the, this WHO and all. We need to take care of especially the vaccination program which is related with the India, with especially reference to the Covaxin and the Covishield. So we are just coming to this point once again, just hold it, right? Just hold it. So the point is what we have concluded so far when we talk about this vaccine, what we have concluded so far, this vaccine, any kind of the vaccine is biological agent or the part of biological agent in killed or inactivated form, and technically the action of this vaccine is like antigen, and after inserting in body it has to stimulate immunity by developing antibody. By developing antibody, right? So the concept of the vaccination is insertion of antigenic agent in the form of complete pathogen in inactivated form, or even the part of pathogen in inactivated form. Then the intention is what? After inserting, your body is going to respond, and that response is what? That response is that response is nothing but the development of the development of the protecting protein, and development of the protecting protein, and this protecting protein is the antibody, is the logic behind, this you know this you know is the logic behind why this antibody is called as the responded protein. Fine. So this is the basic difference between the antigen antibody. This is the basic difference. This is the basic concept of the administration of the vaccine. Why this vaccine administration is required? The intention of the vaccine administration is nothing but to induce corresponding antibody against corresponding antigen. So coming back to the vaccination program of India, and you see here, this is the vaccination program is also called as the immunization program. Vaccination program is also called as the immunization program, and officially 1985 was the year when the national immunization program was started at national level. Clear? And still this is continuing. It is also continuing. Still it is continuing. Still it is going on. And nowadays this universal immunization program consists of vaccination against 12 preventable diseases. This consists of vaccination against 12 preventable diseases, and all vaccines are available free of cost under the UIP, and these are tuberculosis, diphtheria, pertussis, tetanus, polio, hepatitis B, pneumonia, and meningitis due to Haemophilus influenza type B, means hip. Clear? Measles, rubella, Japanese encephalitis, and the rotavirus diarrhea. Right? Rotavirus diarrhea. Clear? And see, so this is what it's an official administration. Official means the administration of the vaccine officially was started in 1985 in India. Clear? And this involved the vaccination against fine? Clear? So again, see, this was the report in 2012. This report was published in 2012. Means this program was started in 1985. So 1985 till 2012, the achievement was 65%. Means 65% of the right. Finally, in 2012, when we analyzed the entire immunization program, so ultimately the total immunization was 65%, right? So again, just to pace this UIP in 2014, we also launched one more program, right? So this was named as a Mission Indradhanush. So to fill the gap, means the ultimate purpose of the Mission Indradhanush was to fill gap under UIP. To fill gap under UIP. This Mission Indradhanush was started in 2014, and the name itself is explanatory. Mission Indradhanush means what? Indradhanush means rainbow, and the rainbow that makes a sense of 7 colors. So this involves vaccination against 7 preventable diseases. So this involves vaccination against 7 preventable diseases, and these diseases are diphtheria, whooping cough, tetanus, polio, tuberculosis, measles, and hepatitis. Alright, 7 mandatory and 2 optional: Japanese encephalitis and Haemophilus influenza. Optional means in selected countries. So this is optional, otherwise these 7 are mandatory. And the intention of this Mission Indradhanush was, see here, the intention of the Mission Indradhanush was vaccination of the children under the age of 2 years and pregnant women. and ultimate purpose was at least to ensure the 90% of the vaccination by 2018. Means this was launched in 2014, and when it was launched with the objective that we are going to achieve more than 90% of the vaccination by December 2018, right? So ultimately, let's see again. The explanatory part would be what? The mission vaccination program at a national level was started 1985 under the head under the head UIP, and this UIP will still continue, right? And then we have analyzed this UIP in 2012, and finally we achieved that the rate of vaccination was 65%. So we launched a special program to fill the gap under the UIP, and that is what it's the mission with the objective to attain more than 90% of the vaccination by 2018, and this involved vaccination against 7 preventable diseases, 7 mandatory and 2 optionals in selected areas, and this involves vaccination, right, of the children under the age of 2 years as well as the pregnant women. Clear? Finally, this mission Indradhanush was further analyzed in 2017, and finally we apprehended that we are not able to achieve this target by 2018. So then to expedite mission Indradhanush, we also launched one more scheme. Means to support UIP, the mission Indradhanush was started, and to expedite mission Indradhanush, intensified mission Indradhanush was started. And again the intention is again intensified mission Indradhanush. So again the vaccination program is same. See, again the vaccination program is same as the intensified. This this vaccination program is again for the 7 preventable disease. 7 preventable disease vaccination program is again for the 7 preventable disease, and here the intention is what? This was launched in 2017, and the target is what here? Here, this is the target. Means full immunization coverage to 90% by December 2018. And here the intention is just to give focus on the urban areas, which was one of the gap in mission Indradhanush. Means that ultimately the purpose is what? To fill the lacuna or the gap of UIP. Mission Indradhanush was started in 2014. To fill the gap of mission Indradhanush, intensified mission Indradhanush was started in 2017 with the intention to ensure more than 90% of the vaccination by 2018. But anyhow, it was not possible. Then we extended the the deadline of this intensified mission Indradhanush, and we launched mission Indradhanush 2.0. We launched mission Indradhanush 2.0. Right, somewhere it is also known as intensified mission Indradhanush 2.0. Clear? And again the intention is it also involves the vaccination. Means the ultimately purpose is also the same. What we have seen, what we have fixed purpose under the mission Indradhanush, that was also the objective of the intensified mission Indradhanush, and again this is also the intention and the aim of mission Indradhanush 2.0. The difference is only deadline. The difference is only deadline to ensure more than 90% of the vaccination. So again here, vaccination against 7 preventable disease. Clear? And here this involves focus on urban and underserved population and the tribal areas. And here we extended this program up to the March 2020. Means the initially target was to achieve more than 90% of the vaccination by 2018. But anyhow, we were not comfortable. That's why we extended this deadline, and as a result of the extension of the deadline, right, this intensified mission Indradhanush was launched as a special operation to expedite intensified mission Indradhanush to ensure to fill the lacuna or the gap of intensified mission Indradhanush. But you must be knowing about last year, March onwards, we were trapped in Corona crisis, and anyhow we were not able to achieve. We were not able to achieve the target of the intensified mission Indradhanush. So that is the reason behind here this third phase of Mission Indradhanush 3.0. Mission Indradhanush 3.0 is launched in February 2021 to achieve 90% of vaccination. To achieve 90% of the vaccination, objective of intensified mission Indradhanush 2.0, because we missed it, which is missed due to Corona crisis. So the target that we missed because of the Corona crisis, just to achieve this target, this separate program was launched in February 2021, and the intention is what? Just to cover the target that we fixed under the Mission Indradhanush 2.0. Fine, okay? Right. So, just quick review of what we have discussed so far. Vaccine is controlled pathogen in an activated form, or even the part of pathogen, or complete pathogen in killed form or in an activated form, or the part of pathogen that is being inserted in body, and the intention of this insertion is to stimulate the immunity, and this stimulation of the immunity is in the term of antibodies, fine. And now we have so many vaccines to protect against infection, like the polio. You heard about the polio. Against the polio, we do have a provision of the oral polio vaccine, or even the injectable polio vaccine, and the intention is what? Intention is nothing but just to develop immunity against polio virus. Again, against the Japanese encephalitis, we do have a provision of the Janvac is the vaccine against Japanese encephalitis. Against, for the dengue, we do have a provision of the Dengvaxia. For the corona, we do have a provision under two different vaccines, that is the indigenous vaccine. One is the Covaxin and the second is the Covishield. In the case of the Covaxin, the complete pathogen, the inactivated form of the complete pathogen is inserted. In Covaxin, the complete inactivated pathogen in controlled form is being vaccinated in body, right? While in the case of the Covishield, a part of spike protein is inserted. That is the difference between the Covaxin and the Covishield. Apart from this, we do have a number of vaccine nowadays. Like you heard about we have also permitted Sputnik from the Russia. We have also one more vaccine is also approved by Indian government, that is the Pfizer vaccine. So that we are not supposed to discuss about, because across the world we have so many vaccines. But we need to take care of only two vaccines which are indigenous, and the difference is what? In one case, complete inactivated pathogen is used, and in the other case, a part of pathogen, that is the spike protein, is used for the vaccination. Nothing else. This is the basic difference. Yes, right? Okay. Okay. So before coming to this, there's a question from Meenakshi. Does polio transfer genetically? Not at all. Not at all. Polio is viral infection. Clear? Polio is viral infection, and it is not being transferred genetically, because this virus is going to affect that part of the brain, that part of the bone which is responsible for the controlling of the muscular development, which is required for that part is responsible for the development of the arms muscles or even the leg muscles. So that part of the brain is being affected by this polio pathogen, right? And that infection is what? That infection is external, right? It is not going to disturb. It is not going to disturb the genetic nature of that particular cell. So the polio is not a genetic case. Wajah Ansari, just changing the name. Target is same. Target, listen, see. Target is same. Whether we talk about I am not talking about the name of one more vaccine, that is the AstraZeneca vaccine or the messenger RNA vaccine of the AstraZeneca, because that is not it. But even I will let you know. Probably in the lecture number 5th or 6th, I will let you know the concept of the AstraZeneca vaccine. You heard about the messenger RNA vaccine of the AstraZeneca, which is not approved in India. Which is already been approved in so many developed countries, but still this vaccine has so many disputes. So I will let you know, but I am not able to make you understand this time, because before this we need to clear the concept of the genetic material, and then I will let you know what is the provision of this messenger RNA vaccine. So now my point is that here, whether we talk about oral polio vaccine, whether we talk about the DPT, whether we talk about the Covaxin, whether we talk about the Covishield, whether we talk about the messenger RNA vaccine of the AstraZeneca, whether we talk about the Sputnik vaccine of the Russia, the intention is same. Intention is same, and the intention is what? Providing immunity against particular pathogen. Right? Now the technique is different. Right? The technique is different. Means the technique for the vaccine preparation is different. That's why, right, one is the Covaxin and the second is what? It's the Covishield. Again, in the case of OPV, and you heard about the OPV and the IPV, the oral polio vaccine and the injectable polio vaccine. Right? We have two different vaccines against the polio. One is the oral polio vaccine that we are putting through mouth, and the second is the injectable, that is intramuscular. Intention is same, and the intention is providing immunity against polio virus. But in both of the cases, the technique is different. That's why one is the OPV and the second is the IVP. So ultimately, the purpose of vaccine is same. The purpose of vaccine is same, and the purpose is what? Just provide immunity against particular contamination. And we could see so many differences among the vaccines, and these differences are nothing but on the basis of the preparational technique, on the basis of the technique which is involved in the process of the preparation, that make a difference among the vaccine. Otherwise, purpose is same, and the purpose is what? It's a target, and the target is nothing but the providing immunity against particular pathogen. As simple as that. Siddhant, which of the two vaccines are better? Do both of them work? See, listen, all vaccines are, means as per the what we have. Ultimately, it is early to make any comment about any vaccine, because we don't have, the sufficient number of data, because the entire world is humankind. But as far as concerned with for the Covaxin and the Covishield, we have seen the sufficient data, right, for both the vaccines. So both are safe. Both are safe. And if some, we have seen so many rumors about the vaccine, the people they have, they took vaccine and they collapsed. So that is not because of the vaccination process. That is because of the vaccination. That is not because of the vaccination process, right? That could be because of the comorbidity condition. Because see what happened in the vaccination process, we are simply inserting the pathogen in your body in the controlled form. So there could be the chance of the fever, there could be the chance of the infection. Clear? And if the person is in the comorbid condition, like the person is prone to the heart attack, the person is prone to the diabetes, the person is prone to the kidney disorder, in that case, there could be the chance of a different, right, lethality. Clear? And as a result of the lethality, because of the other conditions, if the person is going to collapse, so ultimately we are going to put a target like this man collapsed after vaccination. So that is not like that. That is not like that. If the person collapsed because of the vaccination, if it is possible, if it is done. I have seen so many cases, but that was not only because of the vaccination. That was because of so many other conditions. Fine. So both vaccines are same. Both vaccines are same. So don't be apprehensive about the vaccine. It is just to save you. We go and take vaccines. Across the India, they are going to start vaccination process. First May, means as per the availability, they are going to start this vaccination process first May onwards. So all vaccines are safe. Go and take vaccines and stay safe. अरे कोई साइड इफेक्ट नहीं है यार. You people are. Don't talk about the Baba Ramdev. Anant Dhanraj Singh. There is a good question from Anant Dhanraj Singh. Can such vaccines provided to the adult as well as, or these are effective against the children? Listen what happened. Normally, immunity, when we talk about the immunity, the most of the immune systems are already been developed till the age of 5 to 6 to 7 to 8 years, max to max. By the age of 8 years, body acquired immunity against most of the infection. And that's why we do have a provision of the vaccination against, the generic diseases like the polio, like, diphtheria, pertussis, smallpox, chickenpox, all and all. Clear? So by the maximum vaccination is till the age of 5 to 6 to 7. Fine? Because till this point of the vaccination, your body is already well enough to respond against any kind of infection. So maximum vaccination process is done by the age of 5 to 6 to 7 to 8 years. Now this time we are just talking about the corona. Corona is a new case. It's a new kind of the pathogen in ecosystem. It's a new kind of pathogen in ecosystem we never faced earlier. That's why we are talking about vaccination at every stage. Otherwise, that vaccination process we are not going to do. For the polio, we are not going to vaccinate anyone whosoever is more than 5 to 6 years. Right? Maximum means the maximum limit for the polio vaccination is what? It's a 5 to 6 year maximum. And that is the part of Government of India Pulse Polio program. Till the age of 5 years, we should go for the Pulse Polio vaccination to children. Means we are supposed to ensure the every drop of the Pulse Polio program till the age of 5 years, because that is the maximum. That is the maximum age to attain all kind of immunity. Yes, Ayush, we need to remember these 12 diseases. At least not these. See, you can skip these 12 diseases under the UIP, but you are supposed to know the total number of vaccines under this UIP. But in Mission Indradhanush, in intensified Mission Indradhanush, Mission Indradhanush 2.0 and Mission Indradhanush 3.0, in all cases, right? In all cases, there is a provision of the vaccination against 7 preventable diseases, and we need to ensure, we need to remind the name of all 7 diseases, especially under this, Mission Indradhanush and Mission Indradhanush 3.0. Siddhant, still it is what you are asking about the mutation. We have discussed about the double mutant and the triple mutant in the last lecture. Fine. And still, as per the claim of so many companies, अभी मैं कल कहीं पढ़ रहा था कि as per the claim of companies, है ना, the company claims that कि now this vaccine is quite effective against more than 671 mutants variety of the corona. Right? More than 671 mutant variety of the corona. So this is the claim, in fact. Right? This is claim. But we need to analyze, and for the analysis we need to take, we need a time for this entire analysis. So we cannot make comment in nowadays, in this limited period of the time, whether all the vaccines are safe for all mutant varieties of the corona. We are not sure enough, this mutant variety is quite enough, or even we are going to face any more mutant variety in the coming days. Clear? So this could be possible, analysis could be possible after analyzing this, after getting the entire mutant varieties of the coronavirus, and then after the application of the vaccine, whether this vaccine is quite competent enough against all mutant varieties or not. So, the conclusion would be always at the last, but this is not a right time to conclude. This is not a right time to conclude and the comment whether this vaccine is well applicable against all mutant varieties. We need to realize, we need to accept the challenge of the company. We need to accept the face value of the company, and as per the face value of the company, as per the claim of the company, this vaccine is quite competent against the 671 mutant varieties of the coronavirus. Right? So it's a verbal discussion we are talking about. We don't have any established facts against whether it is equally competent enough against all mutant varieties or not. Ashutosh, you just hold your question regarding the mutation of the amino acid. I will let you know once we are going to start this genetic material and the gene expression and the biotechnology. I will let you know what actually the purpose of mutation means, what actually the meaning of mutation in the amino acid. So at this point of time, I am not able to reply your question, but definitely I will reply your question probably in the lecture number 5 or 6 after clearing the basic concept of the genetic material. And then when we are going to follow this concept of the transcription and translation, then I will let you know what actually the concept of the mutation means, how exactly this mutation is going to affect virus. So just hold your question till the lecture number 5 or 6. यार आप लोग बहुत डरे हुए हो. Can a person having both the two vaccines done again suffered by corona? हाँ बिल्कुल हो सकता है. So vaccine is not a guarantee. It's not a guarantee. Vaccine is not a guarantee. So you are not going to be suffered by the corona. Vaccine is a kind of preventive measure. We are just trying to protect you. भाई आप simple सा logic is what? ठीक है. If you are going to counter any gangster act, clear. So you are just wearing a bulletproof jacket. So bulletproof jacket is what? It's a kind of protective measure that you are going to do. Clear, but it is not like that. It is not. No one is going to claim that you have already wear the bulletproof jacket, so you are going to be all right. Probably the bullet is going to crack your bulletproof jacket. The same concept is also applicable in the case of the vaccine. So vaccine is not guarantee you are not going to be affected. Right? You are not going to be affected. But the chance of infection could be least in the case of person, right, once they have both shots of the vaccine. They are less vulnerable as compared to someone, right, he or she has not vaccinated. Clear? Means the person has already vaccinated is least vulnerable to the infection as compared to the person who is not vaccinated yet. Yes, नम्रता, the word antigen is quite confusing. कोई confusing नहीं है. देखो antigen is always, antigen is a protein. आप simple देखो, आपको don't try to confuse yourself in a very simple manner. I'm just trying to make you understand in a very simple manner. Any infection, any infection, any infection, whether it is the tuberculosis, whether it is the bacteria, whether it is the viral infection, whether it is the bacterial infection, whether it is fungal infection, whether it is worm infection, infection is always antigenic in nature. You got infected through coronavirus, it means the coronavirus antigenic protein is infected you. You got infected through the tuberculosis, it means the antigenic protein of tuberculosis bacteria is infecting you. Right? So all infections are antigenic in nature. Simple. It's a very simple. Clear? Infections are antigenic in nature. Right? And these infections are going to infect you. Means these antigens are going to infect you. Clear? And against this infection, your body is going to respond in the term of antibodies. So if your body is competent enough to develop antibody against contamination or against antigenic protein, contaminated antigenic protein, it means you have a good immunity. You have a good immunity, right? Like example, now this is what? It's an infection, and this infection is antigenic in nature. Right? The infection means which part of the infection is going to infect us. Which part of, we are talking about, I am searching for the tuberculosis. The tuberculosis is what? It's a bacteria. So which part of the tuberculosis is going to infect me? It is what? It's the antigenic protein of the tuberculosis bacteria is going to infect me. So this is what? It's the antigenic protein of tuberculosis bacteria. It's a spike protein of the corona. It's the antigenic protein of the Entamoeba histolytica, right? It's the antigenic protein of the pneumonia. It's the antigenic protein of the hepatitis virus. So this antigenic protein of the pathogen is going to infect me, and this is my body. So hitting, this is what? This is hitting my body. And against this infection, if my body is going to develop a responded or protective protein, and this protective protein is antibody. Clear? So if my body is responding well, antibody. So this antibody will not allow entry of this particular antigen. Means I have a strong enough antibody, and this strong enough antibody is protecting me against this infection, which is antigenic in nature. But once I have weak immune system, means if I have weak antibody system, it means what happened? This antigenic protein, means this infection particle or this pathogen is going to insert my body, and this insertion leads to infection. So there is a consistent fight. Every time I am talking to you, I am delivering lecture, right? I am taking water, I am taking tea, I am inhaling. My body is going to get a number of jumps through air, water, and still my body is fighting against contamination. But I am well enough, I am competent enough, I am alright, because I have a good immunity, and my immunity is protecting my body against the infection, and I am alright. Once this infection is going to encroach this antibody, means immunity, right? That condition is what? It's an infection. It's very simple. Fine. Right? So what we have done so far, see what we have done so far. See, this was the slide, right? Connecting you. This was the slide. I'm just connecting you. I told you from the no life to the life, there's a connecting link, and this connecting link is what? It's a virus. And then in this context we have discussed virus, immunity, as well as virus immunity, as well as something about, not 100%. We need to discuss a lot about this coronavirus, but I am not talking this time because I will let you know virus immunity as well as so many things. Fine. Now here, when we talk about this life, the basic unit of the life is what? It's a cell. Clear? The basic unit of the life is cell, and I told you we have two categories of the cells. Like the one is prokaryotic cell, and the second is eukaryotic cell. Clear? So before coming to the cell, means the official topic is what? It's a cell. But before coming to the cell, we need to discuss, right, two more important points, especially in reference to this corona. And let me handle these 2 points, and then coming to the cell. Coming to the cell means the official topic would be the cell, right? But before coming to the cell, let me mention here the two testing techniques of the SARS-CoV-2, and nowadays we are simply using two testing techniques. One is rapid antigen test, and the second is RT-PCR test. This is preliminary test and this is confirmation test. One is preliminary test and the second is confirmation test. Fine. So this RT-PCR test is what? In this case, usually we are taking nasal swab or throat swab. Right? So suppose, example, if I am going to be infected by coronavirus, so see, and that means I am going to inhale coronavirus either through my nose or through my mouth, because it's air transmission. Means I am going to be contaminated, and the way of infection is what? It is through the nose or through the mouth. So this mouth means this nasal swab, this nasal mucus and the throat mucus, it has this nasal and the throat mucus, it has coronavirus. And I told you what actually we are determining in this case. This is what? It's a coronavirus. And I told you this is what? It's a spike protein, and this spike protein is infectious protein. So in the rapid antigen test, we are just trying to determine this spike protein. And I told you earlier, I told you even today, spike protein is what? It's an infectious protein, and the infectious protein is antigenic in nature. So the intention of rapid antigen test is confirmation of spike protein of SARS-CoV-2. That is the severe acute respiratory syndrome coronavirus strain 2. Right? So the rapid antigen test is nothing but it's a testing of. Rapid antigen test is nothing but it's a testing of antigen protein. And this antigen protein is nothing but it's a spike protein. Clear? And the source of this spike protein is what? It's an infection, which moves through nasal swab or even throat swab. Fine? Nasal swab or throat swab. And what about we got touched by, what about if we got corona by touching? क्यों इतना दुखी, इतना परेशान क्यों है मनीषा चौधरी? ठीक है, if you got corona infection through touching, है ना, so just sanitize yourself, as simple as that. Okay, fine. I got your concern. भाई इनका ये कहना है now the Manisha is just trying to ask about कि ठीक है if suppose है ना I touch someone and that man is suffering with the corona. In that case, what is going to be done? Listen, suppose if I am going to touch someone who is corona patient. So suppose if I touch this man and suppose if my hand is contaminated to the coronavirus, so it is not going to affect me. It is not going to affect me because coronavirus is not going to penetrate your skin. So it is quite ineffective. And then we need to sanitize your hand, and after sanitization we are going to disorganize, we are going to dismantle virus. But by any means we touch someone, by any means we are going to touch. Right? This hand means the hand with the coronavirus to the nose, to the mouth. It means what happened? We are simply penetrating coronavirus inside through the nose and through the mouth. Clear? So this virus is going to affect us. Right? So virus is not going to affect you as long as virus is right on your skin or in the skin, but virus is going to affect you by skin, through the skin. If virus is going to be inserted in your nasal or even in the mouth swab, then it will move inside. It will move inside, and then it is going to be infected. That is the logic behind why we need sanitization these days. And the ultimate purpose of the sanitization is what? Ki by default, right? By ignorance, by innocence, we have touched someone is the corona patient. In that case, we should go for the proper sanitization. And the intention of the sanitization is what? Sanitizer has alcohol. Sanitizer has alcohol, right? And this alcohol is responsible for the disassembly of the viral protein, right? This alcohol is when we are going to put protein in the alcohol, this protein will disassemble. It will dismantle. So this is required for the dismantling of the spike protein of the coronavirus. Is the ultimate logic behind sanitization. I hope you understand, Manisha. And then this RT-PCR is the confirmation test. Yeah, 100% nowadays. आप इस बार देखो, इस समय तो we could see so many cases, even the people, they are rapid antigen में they are negative, है ना? But RT-PCR में they are positive. Because आप जो भी यूज कर रहा है, जो भी the condition, you know, the testing kit we are using, है ना? ठीक है, we are assuming that this is accurate 100%, because we don't have enough time to analyze. We don't have enough time to analyze. But even we could not claim that the kit that we are using for the diagnosis of this coronavirus is 100% authentic. इसीलिए we could see the mismatch of the cases, like even if you go to the Dr. Lal Path Lab, one path lab is saying that you are positive and the one path lab is saying that you are negative. Just because of the mismatch, because we don't know the quality of the kit. Possible. Right? Possible. इसीलिए तो that is the reason behind we are simply confirming to the RT-PCR test, because the spike protein, the nature of the spike protein could be similar. The nature of spike protein could be similar for two different pathogens. But here internally, this virus has what? Genetic material. And this genetic material is what? It's the RNA. Right? And RNA is always specific. It is always specific because RNA is genetic material and it is always specific, right? So this RNA is being confirmed in the RT-PCR. Means RT-PCR is what? It's a nucleic acid test. It's a nucleic acid test, or we can say like it's a gene-based test. It's a gene-based test. It's a gene-based test. And even if technically there is no flaw in the process of doing this, a diagnosis, right? Means if there's a technical, suppose if there's a technical problem at the diagnosis part, so in that case the chance could be, the result could be variable. But if technologically means all protocols, they are going to follow all protocols in a very right manner, so we cannot challenge this RT-PCR because this is completely, it's a genetic nucleic, it's a gene-based test. and we cannot substitute, means we cannot falsify the gene based test because it's 100% authentic, right? So see here, what exactly we are doing in the case of the RT-PCR. See, in the case of the RT-PCR, this RT-PCR, we are simply, this is the coronavirus, and we are simply taking this RNA of virus outside. And this RNA, this is what? It's the RNA. And this RNA of the virus is, it's insignificant in amount. Insignificant in amount. We could not justify. It's insignificant in amount. So we are converting this RNA of virus. And we are converting RNA of virus into the DNA. It's a process. It's a process. I will let you know. I will let you know later on. Probably in the lecture number 6 or 7. And there's a gene expression process. Means usually DNA turn its expression into the RNA. It's a natural process. But here we are simply reversing this process. Means the RNA, the expression of RNA will turn into DNA. And this process is called as, and then what happened here? This, and then what happened? This DNA will amplify. Amplify means increasing the number of DNA. Increase DNA number like this. Like this. Clear? Means it's a cycle of DNA. So see what happened. Listen. This is the RNA to DNA. This process is called as what? It's a RT. That stands for reverse transcription. Reverse transcription. Fine? Reverse transcription. Usually there is a term. Transcription. And that transcription is what? That is converting language of DNA into the RNA. But in this case, the process is just reversed. The RNA expression of RNA is turned into the DNA. The expression of RNA is turned into the DNA. This is what? It's a reverse transcription. And then what happened? The amount of DNA is being amplified. Means we are simply increasing the number of DNA. Clear? And this is what? It's a DNA amplification process. And this DNA amplification is what? It is done through polymerase chain reaction. And then by counting this number of DNA, because ultimately this DNA is complementary to this RNA. 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 So here we have sufficient number of DNA to analyze the nature of this RNA, and we named this technique as a RT-PCR. RT-PCR. Right? It's a reverse transcription polymerase chain reaction. Clear? Reverse transcription polymerase chain reaction. Once more, I am going to analyze, I am going to explain once again. It's a RNA, it's a virus RNA will turn into the DNA, This process is called as reverse trans, and then the DNA cycle will amplify like this. This process is done by polymerase chain reaction. Polymerase chain reaction. So ultimately, what we have done in this entire discussion, we have simply conformed this DNA which is complementary to this RNA. So this is what? It's the RNA or gene or nucleic acid based test. And that's why it is conformation test. And sometimes, and then we named it as the RT-PCR. And in the generic term, this is the scientific term. This is the scientific explanation of the RT-PCR. It's a reverse transcription polymerase chain reaction. Clear? But nowadays it is also written somewhere as a real time PCR. RT-PCR. So if you just Google it, in Google it is mentioned as real-time PCR. That make a sense of real-time PCR. That make a sense of, that is the real time is what, that real amount of the RNA will turn into the DNA. That make a sense of real time. So the real time is what? That is the generic term in the term of the amount of the RNA which is involved in the expression of the DNA. Otherwise, technically, scientifically, this is called as what? It's a reverse transcription polymerase chain reaction. हाँ अभी करेंगे आगे. I will let you know. So just hold it. है ना? Apart from this, we have so many tests, but है ना? That is the Faluda test, which is already approved by the CSIR. Apart from this, one more test is already already approved by the Reliance company. So I will let you know. थोड़ा little what is required for the exam. I will explain. Just hold it. No. Ayush amplification processes. Polymerase chain reaction is amplification process, and this amplification process is required only for the DNA. This is not applicable for the RNA. That's why this RNA is first turned into the DNA, then it is further transferred to, then it is further converted to then it is further required to amplify the amount of the DNA, right? So amplification, it's a polymerase chain reaction, is the amplification process only for the DNA. We cannot apply this process in the case of the RNA. Yes? Mubina, why RNA convert into the DNA? Because Mubina, the size of virus is in pico or the nanometer. Clear?